Beth and June, thanks for having me. And thanks to everyone for attending. I didn't see a Virginia in there, but hopefully, if you're there, just let us know in the chat. I did, however, see Tucson, Arizona. That's where I went to medical school. So, so great to have everybody around. And so, the way that I've put this together the last few years is solicited slides from companies that I know that are in the space. And so if they are listening now, I am sorry if I butchered the name of the drug, but I think that the data are clear. And as June pointed out, what an exciting time. I mean, I even from last year, there's new programs in here. So that's really exciting to me. All right, so here's my disclosures. I do work with a number of the companies that we're going to be talking about. And I always like to start from the beginning, just to make sure, in case there's new folks on the call today. So weakness in FSHD is asymmetric. It's regional, it's stepwise. And it typically starts in the face and scapular muscles. Then it tends to involve the humeral muscles, with like the biceps and triceps, and then eventually involves the muscles of the legs. And so actually, if you think about that, it makes clinical trial design somewhat challenging in the sense that, if you think about how you would measure change in your smile, it's pretty hard to figure out. And then if you try and raise your arm over your head and say, well, how do I measure that change? That's also pretty challenging. There's some technology that's developed, but for a long time, this has really led to some challenges in terms of drug development in the space. So here's basically our overview of our webinar today. So on the left, you can see the genetics of FSHD. So as you may recall, you're supposed to have, over 11 of these D4, Z4 repeats. And when you do that, this gene here is silenced, and DUX4 is not expressed. If, however, you have less than 11 of these repeats, which is FSHD1, you end up with what's called hypomethylation, which means that the kind of like unwinding a ball of yarn, and you end up with DUX4 expression, which is toxic to muscles. Or you can have a mutation in a gene that's supposed to wind the yarn tight, which is an epigenetic gene. And there's a test at the end of the webinar. I'm just kidding. And if that's true, then same thing happens. Normal number of repeats, but you still end up with DUX4 expression. And really kudos to the groups in the Netherlands and Rochester that did the yeoman's effort on figuring out the couple of sentences I just talked to you about, because it's really allowed us to precisely target the gene mutation and problem. So here's 5 different kind of ways that you might think about treating this. So the first one is, could you just replace those repeats, which is like gene editing or CRISPR therapy. And right now, there's not even a company that I'm aware of in preclinical development. So we're not going to talk about that, but you should know that would be a potential strategy. The second thing is, that as I said, kind of the DNA is unwound like a ball of yarn that's frayed at the edge. Could you simply rewind it, or what they would call epigenetic silencing of the locus, and prevent DUX4 expression? That's one thing. So there's silencing, and then there's also modulation, which are, in some ways quite related. And then, you don't want DUX4, you don't need it, you're not using it. Could you knock down or silence DUX4 expression? And then last, we'll finish up with, obviously at the end of the day, the expression of DUX4 leads to muscle weakness, muscle atrophy. Could you improve that? So 5 basic ways. We're going to talk about 4 of them today, and we'll use this as kind of our roadmap as soon as I can advance my slides. All right. So as I said, what are the challenges for FSHD? It's caused by this toxic gain of function due to de-repression of DUX4, and it's sporadic. I think, for some of you on the call, you may recall, like, hey, I woke up one day and my shoulder really hurt, and the next couple weeks I just noticed it was so much weaker. That's telling you that DEX4 is being expressed in that area. But that doesn't happen all the time, and sometimes it's not so obvious. FSHD can affect people from birth until old age. So what age do you start treatment? And then there's considerable variability in severity and progression between individuals and within even a single family from generation to generation. If you can think about your other family members with FSHD, that might sound familiar to you. It's asymmetric. So in general, one side or the other is weaker. So, if you're thinking about a trial, do you always measure the left grip or the right grip? These things are challenging. And then on average, I think, maybe a good news for the clinical progression of FSHD, it is a slowly progressive disorder. So, so that means that you may need long trials though. So loss of motor strength on average is about 2 to 4% per year. So I know that feels too fast for those of you living with FSHD, but if you think about it from a drug development standpoint, if you're going to show change and the natural progression is 2 to 4% per year, that tells you you need either a long trial or lots of patients to achieve statistical significance. And then there's no current FDA therapy. So this is really kind of where we started in terms of therapeutic development. The good news is, and a big credit to Dr. Statman at the Kansas University Medical Center, and then Dr. Twill at the University of Rochester, that there's a FSHD clinical trial research network. These are the sites here that you can see. It's really expanded over the last few years. Again, big kudos to the KUMC team for making this happen. And this has a couple implications. These are sites that are really kind of thinking about FSHD. They're preparing for trials, and they're definitely engaged in the space. And then they're doing what are called natural history studies, which is a way of measuring that disease progression. And these studies are so critical for those individuals designing therapies, because tells them, number one, what are you going to measure? Number two, how are you going to measure it? And number three, how long are you going to measure it? Without these efforts in these studies, we wouldn't be talking about the trials I'm going to talk about in the next few slides. So they've done a study called Resolve FSHD, which had 240 individuals enrolled, and that's nearly complete. There's a study called MOVE or MOVE Plus, which is a pretty simple study to do. It's at all of the sites that were on the slide just before. There's an additional part, which is MOVE Plus, which is more involved, which adds muscle biopsy and MRI. But these are both open for enrollment. If you take one thing away from today, is please consider enrolling in one of these natural history studies. This both informs clinical trial design. It also probably starts to engage you with these clinical trial sites. Here's how you might do this. You can contact Michaela Walker at Kansas University Medical Center, and tell her where you're from, and she'll try and get you the closest open site. So, again, please consider participating in FSHD CTRN study. All right, so then let's start talking about drugs. And these are the legal kind. So this is the drug approval process. On average, it takes 12 drugs to be approved. Out of 5,000 drugs that are tested, only 5 make it to clinical trials, and only one of those makes it to market, which is really a little depressing, actually, even during the holiday season. It is my personal perspective that I think those numbers take into account drugs like blood pressure or other things, and I kind of wonder if for these kind of genetic precision therapies, if those numbers are a little bit boosted. But I still think, it's a daunting process. It's pretty hard. Here's what has to happen. Well, first somebody has to come up with a drug, and then they do this kind of preclinical research where they test the safety in animals, and then they work under what's called an investigational new drug application, or IND. And after that point, the drug kind of moves through these clinical trial phases. We're fortunate because FSHD is a rare disease. that oftentimes phase 1 and 2 are combined. So you'll hear me refer to kind of phase 1-2 studies as a single entity, because usually what that means is that they're testing both a single dose and multiple dose, and they're trying to understand the safety of that drug in humans, and they're trying to understand what the right dose is. And then at that point, you end up with this phase 3 clinical trial, which is the, does the drug actually work? Does it do what it's supposed to do? And then at that point, oftentimes there's about a six-month delay, which is what you can see here, after the new drug application is submitted, and then the drug is approved or denied by the FDA. You may hear about this accelerated development review, which is kind of like it's a different process here. And basically what that means is that they are taking data from the phase 1-2 trial and using a biomarker like DUX4 knockdown, for example, and saying, we know that if we knock down DUX4, that drug will end up being, that drug's going to have long-term benefit. So please let us get it approved right now while we're doing our phase 3 trial. So that accelerated approval process does provide earlier access to the drug to the larger population, but it often still requires this phase 3 clinical trial. And this is the U.S. process here. Well, this process here is the same worldwide. The kind of FDA piece of it is U.S., but the process for European medical agencies is the same. And then outside of Europe and the United States, many companies rely on that initial approval from one of those two agencies. Not completely, but we'll try to cover as many countries as I possibly can. So, all right. So who participates in what study? I get that question all of the time. Well, I would say this is the way of thinking about it. So phase 1 trials typically involve very few people, and they tend to be pretty mildly affected, because our first obligation to you is your safety and welfare. So phase 1 trials tend to involve mildly affected individuals. Phase 2 trials, you can see still smaller numbers and typically mild to moderately affected. And then efficacy trials tend to be much larger. They take longer times, and it could be the same population. So, what's the take home here? And unfortunately, I wish this wasn't true, but oftentimes these early clinical trials, number one, just take a very small group of the population, and number two, they take a small number of people, small number, and tend to be a small group. The goal for a phase 3 trial is that both the number of people and the breadth of the population are much larger, while still meeting the goals of identifying the efficacy. So, as I alluded to, particularly in the United States, drug approval process is different for rare diseases like FSHD. So in the United States, there's our Orphan Drug Act, which does provide several forms of regulatory relief. So it allows this breakthrough designation, which means that the company can talk to the FDA more times than before. And it provides a priority review, which is kind of like a fast pass at Disney, where you can kind of jump the line, again, kind of going through the process. So the bottom line is that, like I said, it's probably faster than standard drug development. I'm not sure if I know, and it'd be interesting to do some work to understand, what the rate of success is in rare disease, and if it's different than that first slide I showed you. Okay, can you get the therapy outside of a clinical trial program? Probably not. So for phase programs that I kind of are talking to you about when I call them out as in phase 1 or 2, these early clinical trials focus on safety, and so really the goal is to make sure it's safe before you prescribe it broadly. So I think these, this is an important step. So yeah, so I think that's what I would say. So there are occasionally what they call compassionate use program or open access programs that tend to come pretty late in a drug approval process. Those are highly variable by company. I think if you have questions about this, you can either ask your neuromuscular doctor, or you can ask, the company themselves. They all have some sort of email for patients. All right, so let's get to the meat here. So number 2, epigenetic silence of locusing. So the first program we're going to talk about is from Epic Bio's program, and they have this what's called GEMS platform, which is a process where they use basically, the way to think about it is it takes a virus that wouldn't cause you disease, like a gene therapy type of virus, if you've heard about those in the news. And then they into that virus, they take out the bad virus DNA and they put in this set of this system, basically. So this system includes a couple steps. So it includes a RNA that helps guide the modulator to the DUX4 locus, or the DUX4 region. Then it includes this kind of Cas protein that fuses to the modulator, and then it has these modulator proteins. And the bottom line behind all of this is that the goal is to precisely target or rewind up the DUX4 region. So like I said, the problem is that that region gets unwound, like I said, like a ball of yarn. And when it gets unwound, this DUX4 is expressed and it's toxic. So it would probably make sense that if you could rewind the region, then you wouldn't express DUX4. So that's super cool. And this is just another slide kind of showing you the same basic idea. And this is a very similar to the initial slide, that basically you have this pure D4C4 repetitive elements, and it's what's called hypomethylated or unwound. And essentially this Epi321 will remethylate this region and suppress the DUX4 expression. That is the goal of this particular program. And I meant to say at the beginning, and I'm sorry about all of the technical jargon. Where possible, I'm trying to make it put us all on the same table where possible. So as I said, this Epi321 uses the AAV RH74 virus. This is a virus that doesn't cause disease. It's been used in some of Sarepta's gene therapy programs like Levitus for the treatment of Duchenne muscular dystrophy. So there's a very known safety profile. The one thing I'll say about this, and this will come up again later on in the talk, is that these ones that use these AAVs or viral approaches, right now we think these are one-time treatments because we want to give them to people who don't currently have an immune response to this AAV, and then the assumption is that once we give you this virus, that you would otherwise generate an immune response. So right now we think these are kind of one-time treatments for efficacy. And here's some data to show you what happened with this within Epic Bio for this Epi321. So I'll kind of talk you through what you're looking at. So basically, this first graph here shows you that they're rewinding the DNA about 10% more than what it is right now, 9% actually. But and that when they do that, you can see here's DUX4, here's it's expressed, and it's not expressed, which is great. That's what, again, like I said, you don't want this. And this has been being done in cell lines from individuals with FSHD. And then similarly, when you express DUX4 in cells, you kill them. And this is a measure of whether or not the cells die. And again, you can see that compared to controls, that those treated cells look like they have much less chance of death, reduced cell death in these myoblasts. So this is all quite promising in terms of a treatment really at the root cause of the disease. When you move on from cells, typically people will test mice. And so this is a mouse with a little bit of human FSHD muscle in that. For the sake of time, I'm just going to skip over that part, because otherwise we would spend a whole hour talking about how that was made. But basically, you should know that this is a living mouse with human FSHD in the muscle. And what you can see basically is that, again, looking at no treatment, a low treatment, and then a higher treatment, you can see a reduction in the DUX4 the way we would want to see that, so about 50% or higher, about 60% here. And then same thing, looking at that measure of cell death, again, getting better as you go up to the higher dose. So again, all promising that this has potential to treat the root cause of the disease. And then looking at, okay, so you treat that in the mice, do the mice actually get better? Do they do better? This is looking at strength, really two different ways of looking at strength in the mice. And this is here, you can see the first bar is treating it with the Epi 321, and the second bar is always, it's still a virus, but it doesn't contain the system, the GEM system. And you can see same thing, that there's an improvement in terms of force, really at every dose level that they looked at, even pretty low doses, and that the improvement was significant. So again, suggesting that this might lead to an improvement of the phenotype. So this is EpicBio's planned clinical trial design. I'll draw your attention to a couple things here. So number one, only 9 patients. So again, this is a small study with the primary focus being on safety. So that's an important point. But you can see basically it's a single dose, either at a low dose or a higher dose. And then, over the course of a year, there's visits about every 3 months looking at muscle biopsies, muscle imaging, and then, walking speed and other clinical outcomes. So this is a planned phase 1/2 trial. I don't think we know exactly when that trial is going to start, but they've made it all the way up until this point. So hopefully sometime soon. All right, keeping on this track of kind of rewinding the DNA, or epigenetic methylation. If you're confused about epigenetic methylation, don't worry about it. A number of medical students are as well. It's a pretty complicated topic. Here's the bad news. So this is, I have to put this in there, that a drug called losmapimod acts on this kind of methylation piece. It's a MAP kinase inhibitor. And many of you may have participated or be aware of that Fulcrum had a positive phase 1/2 trial. They then moved on to a phase 3 trial, and that trial was negative, and that program's now been discontinued. There's no change in a measure of reachable workspace, which is like how well the shoulder moves, or shoulder strength compared to placebo. And actually, one of the things that's important for us all to keep in mind is actually the people who got the placebo or the sugar pill actually got better, over the course of the trial as well. Not as much as what we would expect and have seen in other clinical trials, but it's just really remembering the importance of that placebo group, and the placebo effect. I think when you're getting something that you think is working, people got better. Okay, so this is actually the meat of the talk. We've got a number of programs here. Don't want DUX4, don't need it. Let's talk about how people are treating this. Well, the program that's definitely the furthest along is the Avidity program here, AOC 1020, or Delbrax. I can't say the full generic name. And this is basically, it's a 3-part compound. So one, it's a silencing RNA to the DUX4s. Like I said, it's a piece of RNA that will degrade the DUX4 expression. And that is bound to what's called a transferrin 1 antibody. So your body doesn't naturally take up RNA. It looks like a virus. And so people need to do something to help your muscles take the RNA up. And this is one way to do that. So that's using the transferrin 1 receptor, which is in charge of metabolizing your iron, to deliver that DUX4 into the muscle. So clearly in both a mouse model, there's broad delivery to muscle, and there's clear efficacy using the same kind of preclinical model. The phase 1/2 trial presented their initial results right around the FSHD 360 in June. I was impressed. I think many of you were as well if you saw it. So basically, this is a randomized double-blind placebo-controlled trial. It involves adults with FSHD. It has a 12-month follow-up. There are biopsies to see whether or not that DUX4 is being knocked down, and the company was able to show that they were able to. So far, it's been shown to be fairly safe and well tolerated in the data that's been released so far. So, we'll continue to follow along because they're still in the process of kind of getting all the data from from both the lower and the high dose cohorts at all of the time points. But I think their initial data suggested that they were getting their drug to the muscle, that they were knocking down DUX4 the way they wanted to, and that actually had some benefit in terms of strength and function, which is great. Here there is, I'm going to skip past this, do this biomarker cohort because I got these slides one out of order. So this is basically the other thing that was in that first phase 1/2 clinical trial was that there was a biomarker in the bloodstream that was suggested to be related to DUX4 expression. Now, even though I really enjoy taking muscle biopsies, I recognize most of you out in the call prefer not to have them. And this would be a huge advance if we were able to take a blood draw and see whether or not we were able to knock down DUX4. So based on their initial findings, which were, I think, a bit of a surprise, they added this additional cohort called a biomarker cohort. And that cohort is being enrolled at this moment with the dosing 2 mg per kg every 6 weeks. And it's essentially to prove whether or not the, that biomarker still works in a bigger group. And the same key exploratory biomarkers or exploratory objectives. So the biomarkers, measures of clinical activity, and then patient and clinical reported outcomes. And so right now, this is kind of where they're at with this 2 mg per kilogram dose. And I think, there's still this a plan to have a future cohort. I think that the timing is to be determined. Same idea. This is from Arrowhead. So this is a phase 1/2 trial looking at a very similar approach. So this is same thing. So it's an RNA interference approach designed to knock down DUX4. And this is basically the way that works. And here, rather than using the transferrin 1 receptor, they used a piece of protein that with the same basic idea that they're intending to get that into the muscle. A very, very similar approach. This trial is just getting going now. So we don't really have any data about the outcomes of it. You can see that this trial is not being conducted in the United States, but really initially started in New Zealand and Australia, but has since gone to Canada, Thailand, and the European Union. Here, same thing, there are muscle biopsies. The goal is to understand the concentration of the drug, whether or not it knocks down DUX4, to look at muscle fat fraction, and to do the functional testing that you can see there. So no data. This trial is actively enrolling in those countries that you can see there. All right, just moving right along. This is miRecule's program. So it's a little bit different. Here, this is an antisense oligonucleotide. Oh, and I'm sorry about the print here that it got duplicated. So this is an antisense anti-DUX4 that uses a nanobody to help it have uptake. And this is a partnership with Sanofi, which is, I think, will help in terms of clinical development and certainly within commercialization, given the resources that Sanofi has to bring to bear to this. And they're going to collaborate on this up to the investigational new drug. This is basically their the antisense oligonucleotide or DREAMer can both silence DUX4 and inflammatory gene targets. So again, can work both at the DUX4 itself and prevent the DUX4 protein, but it also actually is a little bit more broader and can look at inflammatory RNA. So for those of you, we didn't cover this, but for those of you who are not aware, when you get muscle damage, you get natural inflammation. And so that can lead to secondary problems. And this is a mouse study here looking at this as it stands right now. So this is the compound here. So this RNA therapeutic, the nanobody injected into the mice. And you can see the expression here. So it's getting good uptake into the muscle. And then a good uptake into the muscle, and it appears to have a benefit there as well. So, and then the last one in this particular section is from Kate Therapeutics. So they have an FSHD program here. They're going back to using this AAV technology. So this is a virus. It's actually their virus that they use is a proprietary virus that's been made by the company using what's called a MyoAAV. And so the goal of that particular virus is that it has the ability to de-target the liver or some of the side effects that you typically see with the AAV viruses, and is better able to target the muscle. So, the analogy there, or the idea is that you could potentially use a lower dose and potentially have more efficacy than a naturally occurring AAV. So they're using a muscle-specific promoter. And then here they're using the same basic ideas. So it's a microRNA, again, designed to knock down DUX4. And the news, if you were following over the last couple of weeks, is actually that this company was actually acquired by Novartis. So I don't think we have details about how that affects their timing or plans, but certainly Novartis, again, has more resources to bring to bear to move that program forward should they choose to. This is the data from their proprietary virus. So this is the virus that you can see here compared to some naturally occurring viruses. And this is looking at the skeletal muscle and where you want it, and the liver where you don't. And so what they're showing is actually that they do get it where they want it, right? So they get more into the skeletal muscle and they get less into the liver. All right, we are moving right along. So we will have plenty of time for questions. The last program to talk about is the modulation of muscle atrophy. So there has been a long-standing interest in a gene called myostatin, a protein called myostatin. And you can, if you're like super into this, you can Google myostatin cows or follostatin cows, because if you do this, what myostatin does, if you overexpress it, it causes a loss of muscle mass. And if you inactivate it, you can actually increase muscle mass. And so it's one of these like basic human genes that's designed to regulate the size of your muscles. And so it would make sense that in a disease like FSHD, where you'd have problems with muscle size, that maybe if you worked on this, it would generally be able to provide some benefit. So in a mouse model of FSHD, inhibiting the myostatin increased the muscle mass and strength. There have been other myostatin type trials in FSHD that to date have been negative, but the one we're going to talk about is the Roche program, which is this right here. So this is a protein that's GYM329 that's designed to block the function of myostatin with the goal of course, increasing muscle growth and function. So it binds to this kind of precursor of myostatin and stops it being converted into active. And of course, the goal is generally that the muscles get bigger and have and stronger. As I mentioned at the beginning, there have been prior myostatin programs. And so this particular program has a few additional technologies to help it do a better job, basically, of binding to that latent myostatin and getting rid of it. And you can see here it both can bind to it multiple times, and then it can clear it more quickly. It's the idea behind it. And this is the phase 2 multi-centered clinical trial sponsored by Roche called MANOEUVRE. And like all the other phase 1/2 clinical trials, this is designed to test the safety and tolerability of this, and to look essentially to start to look at the early efficacy. So it's about a year-long study in individuals with FSHD, and it's fully enrolled at this point. And this is the schematic. It's half the people get the drug, and half the people get placebo. And like many of the other clinical trials, this only at this point involves adults with FSHD type 1 or type 2 with moderate severity and able to walk. So these these inclusion and exclusion criteria, even though we've only touched on it briefly in some other programs, are pretty typical for these early phase 1-2 clinical trials. And the exclusion criteria are also pretty typical. Basically, they have to otherwise be pretty healthy. So they're looking at muscle volume by MRI. They're looking at the safety, and then they're going to start to look at some of the strength measures. And like I said, it's got 10 sites across these countries. It is complete. So hopefully hopefully by next year, this time we'll be able to have some of these results from some of these other trials. So I think that's a whirlwind tour through all of the open or soon to be open programs across FSHD. Really an exciting time, lots of different opportunities. What can you do right now? You can enroll in a natural history study like MOVE and contact Michaela Walker. That really helps design better clinical trials and prevents failures. Because the worst possible scenario in my book is that you have a drug that you think works and then you do a clinical trial and it doesn't work because the trial wasn't designed right. So I think that's really an important part. There are registries, including the FSHD Society has a new registry that allows you to participate and get more information, and then stay informed on drug development programs. There are many of these that are moving forward. So, I think which trial should you do? Because you're looking at that like, gosh, I could do this one or that one. There's so many choices. Well, the first thing I say is, I think if you're interested and you understand the risks and benefits and kind of work through that process, I would do the trial that you could get into, that you and your physician thought were the best suited for where you are in your disease course and what your risk preferences are, et cetera. So, last point here, this is really just the beginning. I've made the point a couple times, it's here on this slide. The trial populations are usually narrower than the general population. In fact, they are always narrower than the general population. And I certainly have people in clinic, and maybe some of you are out there thinking, well, I don't fit any of those things. Well, that is something that both that us as the clinicians, you as individuals living with FSHD, and the FSHD Society are working really hard to prepare for, because we know that when drugs get approved, it's often kind of it's a time-consuming process where we have to work with insurers both on payment models, and then thinking about, how do we get this drug approved for you? And they said, well, you weren't eligible because you weren't in a clinical trial. And you're like, well, but I would still benefit. And so we know that there's a lot of work ahead of us. And I think we need to be prepared that we don't sit there and say, well, now we got this drug approved, we can just go home and relax. We say, well, now we've got to do the real work to kind of get the drug access to these really interesting and potentially transformative therapies to all people who would benefit from it. And the last thing, of course, is that once we do get multiple drugs, I hope you can see there's 4 different steps that people are working on. So it might make sense to work on step 2 and 3 at the same time, or consider combination therapies. So I guess, unfortunately, I can't retire this year. There's plenty of work to be done, but exciting to see it move forward across the board. And so I just want to thank you on behalf of our center here. That is my last slide. And I think I have time for questions.
Yes, you do have time for questions, and we have a lot of them. Thank you so much, Dr. Johnson. And I don't know if you want to maybe unshare your slides so we can
I can unshare it. Yep.
There we go. Great. So I will jump right in because we have quite a few questions. I hope we can cover many of them. So many people are very interested in the Avidity trial. They're wondering if there are future cohorts that are being planned that will open up, or are they already full? Like, do you have waiting lists and you have more than enough?
Yeah, I mean, I think so. I'll try and take that in a couple of points. So yes, I would expect them to have an additional trial cohort that would be available to enroll. My understanding at this exact moment is that the current cohorts are either filled, or people have been committed to the slots that are remaining to be enrolled. So I think right now, unfortunately, if you're just thinking, gosh, I want to do the Avidity trial. I don't think that there's an open core at this moment. But I think what helps me, and I know helps many of my colleagues, is if you reach out and say, hey, can you put me on your list? I'm really interested in that. And that's what we do is we, when the new cohort will open, we look at our list, we look at who might be eligible, and then we kind of go from there.
There's a question about the clenbuterol trial. Is that has that activated yet?
So I think they're still working through the process of getting started, and a good question about that. That is something that would work very similar to losmapimod. So again, trying to kind of wind that DNA back up. It uses a lot of the same ideas, and that trial will be recruiting, I think, 3 sites, maybe a few more. And I would hope in the new year. So.
Okay. There are several questions here. I'll try to package them into one question regarding the Fulcrum trial. And it was notable that the placebo group didn't decline or maybe even slightly improved. And so is that just a matter of bad luck? Is it that the placebo group started all exercising like crazy because they were hoping to see an effect from it? Can you theorize about what reasons why Fulcrum decided?
Yeah, I mean, I think there's 3 important points from that trial. The first is that, these are clinical trials. And I know in our head, and you're looking at this and you're like, this should definitely work. It's 100% going to work. Well, they are trials, and unfortunately they're experiments. And when they're well designed, as the Fulcrum trials was, sometimes they say the answer is no. This is not a drug that's going to be effective in individuals living with FSHD. And it's a terrible answer, but it's I think helpful when you design a trial and you're able to get an answer that is as clear as what Fulcrum got. The second point is that the placebo effect is well known. I know many of us will have a little bit of a placebo effect on January 1st. We make our New Year's resolutions and put a little spring in our step. I think when, there's something about kind of, being a clinical trial is kind of exciting. You think that you're doing something that will better your health, which hopefully it will. And people tend to take better care of themselves and maybe exercise a little bit more. And so I think it's just a known effect. And I think if you're not in a trial right now, it's a reminder that a positive attitude on outlook on life can also can help your disease even a little bit. So. And then the third point is just, I think sometimes people say, well, why do we need a placebo control in the first place? Well, that's the answer, because we know that people perform differently if they think they're in a clinical trial than in, say, a natural history study.
There's a question about Avidity and their potential biomarker, wondering why they haven't disclosed what the biomarker is. Do you think they are protecting, they're patenting it or have intellectual property on it? And
Yeah, these are good questions. I mean, I think I can't speak for Avidity, and I haven't asked them personally, but I think, some reasons. Number one, I think that they achieved an initial signal of it, but not proof. So I think it's fair to allow them the time to understand in that cohort C, really test whether or not this is something that can be useful. And I think that's to all of our benefits, because if you sometimes you get a kind of interesting answer in the first paragraph or 2 of a book, and you're like, you go out and you're saying, well, this is what it's going to be, and then everybody starts saying the same thing, but then you get to the end and you're like, actually, this wasn't quite true. So I think one thing is just, they're still going through the process to validate it, and people don't ask me to give my results before I feel comfortable giving them, and I think that's probably true in that case as well.
makes sense. So we'll now switch to Epic. I think they're. Can you hear me?
You froze for a second, but you said Epic.
Oh yeah.
June, maybe if you close your video, it might be better for your Wi-Fi. There you go.
Okay. I have some questions relating to Epic, which is now called Epic CRISPR. I think the company renamed itself. wondering, do you know what the timeline is for them to complete this, their phase 1 study?
Yeah, so I think they, well, I can tell you that they haven't started yet. And typically for these early gene therapy type clinical trials, there's a waiting time, a month to 2 months between trial participants. So I would say even with 9 patients, which I'm sure they'll be able to find, you would figure that enrollment would probably take over the course of about a year or so, maybe a little bit longer. Just speculating on my part. And then the trial design suggests that it would be, people would be followed along for at least a year. So, if you kind of add that up, that's saying that by the time they start, it would be maybe up to, 2 to 3 years before they would have full results. Typically companies will give us results in between the beginning and the end, but obviously that's up to their discretion to provide that information.
Here's an interesting question, and our audience, they are so up on the science here. They note that because DUX4 is involved in early embryonic development, would EPIC's treatment be something of concern if somebody received it before they start having children or want to have children?
Yeah, I think kudos, first of all, kudos to everybody out there who knows as much the science as I do. This is true. So DUX4, to date, the only meaningful role DUX4 has is in like the 4-cell stage of embryogenesis, so very, very early on. And I think the answer to that is unknowable at this moment. And I'll just kind of speak broadly, even beyond Epic Bio, is that typically what companies will do is that they'll do the first round of toxicology studies, which allows them to give the drug to adults. And then, which is like, that's what you want, that's what they want. We all want to start the trial at some point soon. And then typically later on in the trial, they will go ahead and do more toxicology studies, particularly looking at fertility and pregnancy and that kind of thing. And those tend to occur later on in a drug development process. So I would say for those people participating in the clinical trial, obviously they sign something saying that they won't get pregnant during the course of the clinical trial. I think for a therapy like Epic CRISPR, where it's a one-time therapy and likely to stick around, I think there's probably some additional considerations there. But as it stands right now, the answer to that is unknowable because it's not really clear whether or not the drug would be present. You'd have to know whether or not the drug was present in the embryos or sperm at the time of embryogenesis, which is really the main question. We don't have that answer. Good question, though.
Yeah, it is. I have some questions now about muscle biopsies. One person said they had one early on, 1980s, and it was excruciating, and it's why she has not wanted to participate in any trials that involve a biopsy. Can you talk about what are the biopsies like that are done today for the trials? Are they similar to these open biopsies that were done back in the day?
Yeah, no, they're not. The biopsy technology's come a long way. And it's important to remember, so the biopsy that that person had was probably done for diagnosis. So for diagnosis, they take a big piece, they take it out to look under the microscope and everything. And for these biopsies, we know what the diagnosis is, first of all, and we're not really looking at how the muscle looks under a microscope. And so they take just a teeny amount of muscle, enough to test for the RNA and protein and usually a few other things. And in that situation, most of these programs use some sort of fine needle biopsy, which is, like an argon 14 gauge biopsies. It's a little bit bigger than a blood draw needle. Now, it's still not the most comfortable thing you've ever had done. So I don't want to shortchange the biopsy itself, but it's substantially less painful when done correctly. Compared to a diagnostic biopsy that you would have had in the 80s. And we use lidocaine, so.
Okay. This is a curious question. What if you are one of these mosaic cases? Would you qualify for a trial? Would you be considered a typical patient or different enough that?
Yeah, I think that is a tricky question. So, and we didn't go into a lot of detail, but most of the trials will require some degree, some measurement of the FSHD. Not all FSHD would qualify for all of those trials. So sometimes people only want like under 8 repeats or some degree of methylation. And so I think that that would be something where if you met every other inclusion criteria, what we would do at our sites, and I'm presumably your physician would do as well, as take the result back to the study sponsor and say, hey, what do you think about this? And then, it's probably a case by case basis.
Okay. We have a couple of questions from people in India wondering if there is, I think I'd say, is there a prospect for there being a trial site in India in the future?
That is a great question. I know in some of our research networks that are affiliated with the FSHD CTRN, we have been looking at adding some natural history sites in India. And I think the FSHD CTRN would likely be amenable to that as well. Typically, that kind of early natural history sites is helpful because it kind of lays the groundwork and people get familiar with the disease, and you can put on that map that companies might look at to say, where are my trial sites? And so the thing that you could do would be to talk to your physician in India and say, hey, are you interested in doing this? And then they can reach out to me or Michaela Walker or Jeff Statland, and we'll make sure that they have a conversation.
Great. So in our last few minutes, these are kind of interrelated questions. One is regarding whether with a rare disease that has no pharmacologic treatment right now, whether new therapies, experimental therapies for FSHD might fit the criteria for the Right to Try Act. And then related to that is, since clinical trials have fairly restrictive criteria, there's a real concern that payers may not cover therapies for people kind of the more larger, more diverse FSHD community. What can we do now to either encourage more diverse trials or pre-approval access programs?
Yeah, those are great questions. So I'll make a couple points at the top. Number one, the FSHD Society is working really hard on making sure that every individual has access to these therapies when they're approved, either by FDA, EMA, or a related regulatory agency. And they have a project called Project MANOEUVRE that is on their website. So I don't have time to cover everything, but definitely please take a look at that. That's one point. Number two, I think we just need to take a deep breath and say, look, the very first objective is to get that drug approved as quickly as possible. And so there are costs to cost us as a community when we make that decision, and that excludes people from the clinical trial. And that definitely sucks. And I think I feel that. I know the companies consider that when they're thinking about their clinical trial. But the first thing that can't be jeopardized is the ability to have a successful phase 3 clinical trial. Because if phase 3 trial is negative, then nothing happens. I think it's, as I kind of put on my last slide, that first successful phase 3 clinical trial is the beginning of the journey, not the end. And I know that the companies that I've spoken to and the clinicians are committed to continuing to do additional studies that would continue to expand the access to these therapies. It's just not ever fast enough for either you or me. So I think that's something to kind of consider on that point. And then the last thing around the right to try, I haven't been broached for that, and I think that would be something where you might consider engaging or reach out to the patient advocacy representatives at any of these companies. They all have one on their websites, Typically those would not, you would not be eligible for that type of program until a trial's in phase 3. So right now, I don't think, in fact, I know none of the trials are in phase 3, so it's not a 2024 issue.
Okay. Let's see, there's so many questions here. People ask, okay, are there any supplements that can be helpful in the meantime, such as creatine, vitamins, antioxidants?
Yeah, supplements, no supplement has ever been shown to have a benefit in terms of disease progression, but they are typically shown to be safe. And then the one thing I will say is exercise. So regular exercise, as you actually saw in the Fulcrum phase 3 trial, does have a benefit. So certainly keep that in mind.
Oh, and here's an interesting other question. For people who've had the scapular stabilization surgery and they have, better mobility in the upper body, does that disqualify them from a trial that uses something like space as an outcome measure?
Yeah, that's a good question, and that's actually, I would say that's highly variable based on the trial. So that's that I would say is really kind of a case by case basis.
Okay, great. Well, we have reached 2 o'clock. And many more questions. So we hope we'll have you back again to give another update and continue to report on the progress of our field. But in the meantime, thank you so much. And I will turn it over to Beth for some announcements.
Yes, thank you everyone for joining us today. Special thank you to Dr. Johnson for this amazing update, answering all of our questions, and most of all, Dr. Johnson, for all the work that you are doing for and on behalf of our FSHD community. We are so very grateful and very lucky to have you in our corner for sure. Dr. Johnson mentioned our registry platform, and I'm going to put a little link in the chat here. So if you haven't already, please check out Better Life FSHD. This is a groundbreaking research platform that is designed specifically for the FSHD community. It provides a one-stop hub for tracking, for information, for research contribution. It's really a great platform. So I encourage everyone to get on board to experience everything Better Life has to offer. It's and all the ways that you can contribute to research. It's a really cool platform. So the link is there in the chat. Our next FSHD University webinar will be Thursday, January the 16th. Our guest speaker is Jenny Holy, who will speak about mindfulness for reducing stress. And so just be sure to visit our website events calendar for all of the upcoming chapter, wellness, educational events. And on behalf of everyone here at the FSHD Society, we wish you all a joyful, peaceful holiday season and a very, very happy new year. And we'll see you next year. Take care, everybody.
Happy holidays.
Happy holidays.
Thank you.